One of the new projects in the laboratory is really exciting. It may make some considerable changes going forward. What we've done is essentially use a kind of tricky, interesting way to clone a little piece of DNA, meaning being able to move a little piece of DNA that has random sequence. What happens is that when you install this into a plant, it produces a random peptide, a random small little protein that sometimes can affect the way a plant develops or the way a plant behaves. Once we identify what these small peptides are, it gives us an opportunity to potentially create new classes of growth regulators. Maybe new peptides that you could spray onto a field to control when a crop would flower, or how it would senesce, how it would die off or dry out. Maybe you could change herbicides by using peptides, which are much safer for the environment potentially. So there's a lot of things that we could do with these technologies. The interesting part is that the possibilities are endless. We could spend all day, every day for years going through libraries and libraries of peptides and never run out of something to test. My laboratory also is interested in how you make fruits and vegetables taste better. And one way to do this is to use really cool tricks with genetics, analytical chemistry, and genomics to identify the genes that play roles in controlling the trace compounds that are there that contribute to the flavors of fruits and vegetables. And so currently my laboratory is identifying in strawberries all of the different genes that control traits that make them taste sweeter, make them taste more fruity or maybe floral. And it's really interesting because when you get all of those genes in the right place, just through traditional breeding, you end up producing a fruit that tastes much better that consumers want to buy.